Hello students. So today we are going to do the next experiment, and that is experiment number 4, which belongs to the next chapter, and that is next component, that is carbohydrates. If you can recall our first class of food chemistry, we discussed various components of a balanced diet, and one of the components whose main role is to provide energy, as you can see, is that of the carbohydrates. So in the picture which is given over here, you can see several sources of carbohydrates, different type of carbohydrates. So let us see what we are going to do in the experiment today. uh, our experiment today, the objective is to identify different type of carbohydrates. I'm sure you must be remembering your earlier courses of maybe general organic chemistry or organic chemistry 1 and 2, and there you may have identified several compounds or different functional groups. So today we are going to perform different type of tests on several samples of carbohydrate. Okay, given samples of carbohydrates, and then had it been in our lab, if we would have done, then in that case different food substances would also have been used in order to identify the various type of carbohydrates. So as I was saying, carbohydrates main role is to provide energy. In fact, many times it is considered to be an instant source of energy. Instant source of energy. I'm sure you may have experienced it yourself. Sometimes when we don't eat the breakfast, we just grab a cup of tea or coffee with milk, sugar, and it uh, like it is sufficient for maybe half the day, right? So that sugar over there provides you with the instant energy, and you are ready for work, right? So as I was saying, carbohydrates are considered to be instant source of energy amongst all the food components. I'll mention again that this is the only component, probably the only component of food whose main purpose is to provide energy. Let us start with the main type of starch you have been consuming, like maybe since you were a child or an infant. One of the main starches that we consume is in the form of starch. I'm sure everybody would have heard the name of starch, and this starch is usually obtained from plant sources. So different type of plant sources. If you are eating rice, if you are eating wheat, if you are eating, let's say, maize or corn, if you are eating potato, all these are good sources of starch. Okay, so starch is probably one of the most you can say dominant or popular type of carbohydrate, and it is obtained from the plant sources. In fact, this is, you can say, in amongst the food item, this is the most important source of most important type of carbohydrate that we are consuming. In our body, we have another similar type of carbohydrate, and that is glycogen. Okay, so we will see just after a while about the similarity between starch and glycogen. So starch, glycogen, and there is another one I would like to mention over here, though we may discuss it later, and that is cellulose. Okay, so actually these three are the polysaccharides, means the most complex form of starch. Now, if we consume starch, let's say in the form of rice or wheat or corn or potato, this starch is metabolized in our body to the simplest form of carbohydrate, and that is glucose. You can see over here glucose. So glucose it's one of the simplest form, and I would say the most common form of carbohydrate. Okay, so it is usually the polysaccharides which I was mentioning, like starch for example, is broken down by our body into the simplest unit of carbohydrate, that is glucose. Okay, so glucose is a monosaccharide basically, and several monosaccharides join together to give rise to a polysaccharide like starch. Okay, in fact I would like to mention here the type of bond that exists between monosaccharide and monosaccharide, several monosaccharides giving rise to polysaccharide. It is called glycosidic bond. Okay, glycosidic bond. Now once the starch is broken down into glucose, what happens is that the carbohydrate which is formed upon oxidation, it provides energy to the cell, and this in turn is responsible for our daily metabolic activities which are happening in our body. So it is basically these monosaccharides, you can say, which are the building blocks of the polysaccharide, or you can say that they join with each other, resulting into more complex molecules, like several units of glucose join together to give a polysaccharide. However, there are certain other class of uh, carbohydrates as well, and we will talk about it in a while. Before we start with the classification of carbohydrates, you can see over here, carbohydrates will either have an aldehyde group as in case of glucose, or a keto group as in case of fructose. Okay, so generally the aldehyde containing group together, they are called aldoses, or such sugars are called aldose sugars, and the ones containing keto group is called the ketoses. Okay, so the structure of glucose, if we see, it contains an aldehyde group, whereas a keto group is found in fructose. Fructose is the sugar which is found in fruits and is actually considered, maybe I can write it here. So fructose is basically a ketose sugar and is the sweetest known sugar. Okay, sweetest sugar. It contains a keto group. Okay, so sometimes it is also referred to as fruit sugar. Let us now see the classification of carbohydrates, how carbohydrates are classified. You may have heard the term simple sugar and complex sugars, or you may have also heard the fact that it is highly recommended that we should use complex carbohydrates in our diet. So let us understand how do we classify simple and complex, and then definitely, but then we will understand their function or their role in the metabolic activities or the chemical reactions that they show. But before that, let us try and understand the definition of carbohydrates. So how do we define carbohydrates? Carbohydrates are said to be polyhydroxyaldehydes or polyhydroxyketone, or any substance which on hydrolysis gives polyhydroxyaldehydes or polyhydroxyketone. I will just draw the structure of glucose over here to re-emphasize on the definition that was just told to you. So polyhydroxyaldehyde, what I am drawing right now is the structure of a glucose molecule. You can see several OH groups in the structure. So the other side is hydrogen, this one also hydrogen, and down there is CH2OH. Okay, so what you can see over here is are several hydroxy groups, and the main functional group you can see is the aldehyde group, hence the name polyhydroxyaldehyde. This is the structure of a glucose molecule. The structure of fructose molecule, I will just draw it now. So fructose has keto group, okay, and so again it contains several OH groups. On the other side it will be hydrogen, hydrogen, hydrogen. So again you can see several OH groups, polyhydroxy ketone. So there is a ketone functional group, and this is the structure of fructose. Okay, so this is fructose. So as I was emphasizing on the definition of carbohydrates, they are defined as polyhydroxyaldehyde or polyhydroxyketone, or any substance that on hydrolysis gives us polyhydroxyaldehyde or ketone. Based on this definition, let us now classify carbohydrates. So as you can see, carbohydrates are classified into two types, saccharides and polysaccharides. Saccharides are many times considered to be sugars, and polysaccharide as the complex sugar. Now, these polysaccharides, since we already started our discussion with starch, these polysaccharides are macromolecules. Macromolecules. Big in size, okay? So big, so much so that our taste buds are not able to absorb these molecules, and therefore they appear to be tasteless. If you have never experienced, take a pinch of flour, wheat flour, rice flour, corn flour, anything, and put it on your tongue, in your kitchen, and you will experience that they don't have a taste of their own. Okay, they are completely tasteless. Why they appear to be tasteless is because they are such big molecules that our taste buds are not able to absorb. Now let us compare it with the saccharides. Okay, so saccharides are definitely smaller molecules than polysaccharides, and also most of these saccharides appear to be sweet in taste. Why they appear to be sweet in taste? Because they have smaller size as compared to polysaccharide, and therefore our taste buds are able to absorb these, and thus we experience the sweet taste of saccharides. In fact, that's why they were named as saccharides, which means sugars. You may have experienced yourself. If you take a plain piece of bread and start chewing it, initially you may not get any taste, but if you keep on chewing it for a long period of time, there will be a sweet taste that develops at the end. The reason behind is bread is made up of starch, which is a polysaccharide. So initially you don't get a taste. But then this starch, there is an enzyme amylase in our saliva, and this starch is broken down by amylase into glucose molecules or smaller molecules, you can say. And once it is broken down into smaller molecules, our taste buds are able to absorb it, and that is when we start getting the sweet taste. So now let us see what is the classification. So as you can see over here, they are classified into mono, di and oligosaccharides. Monosaccharides, what are monosaccharides? I would say they are the simplest form of carbohydrates. Okay, in fact they are also called simple sugars. So they are the simplest form of carbohydrate. Means you cannot hydrolyze further a monosaccharide into another carbohydrate. It's like a basic unit of carbohydrate. Okay, like we have atoms as the basic unit. So likewise here you can say saccharide unit, one saccharide unit or monosaccharide is the basic unit of carbohydrate. So they will not further hydrolyze or break down into any simpler carbohydrate. These are the simplest carbohydrates. When two such monosaccharide units are joined together by which type of bond? It is glycosidic bond. Okay, so when two monosaccharide units are joined together by a glycosidic bond, it results into a disaccharide. You can see over here two monosaccharide units. So when you will do hydrolysis of a disaccharide, it will give you the two constituent monosaccharides. Likewise, if you see oligosaccharides, oligosaccharides are 3 to 10 monosaccharide units joined together. So it can be same units, it can be different units, but starting from 3 up to 10, if they are joined with each other through glycosidic linkage, then they are said to be oligosaccharides. However, if the chain length increases more than 10 units, then we consider it as polysaccharide. However, in reality, you would see that these polysaccharides are big molecules containing hundreds to thousands of units of the monosaccharide. Okay, so it's not just more than 10, but for the sake of classification, you can say more than 10 units, we start calling them polysaccharides. 3 to 10 oligosaccharides, 2 we call them disaccharides, and if there is just the simplest saccharide unit, we call it monosaccharide. Let us see the examples that we are going to, or you can say the samples which we are going to use today belonging to each category. So from monosaccharide, the samples which are available is glucose and I think fructose, maybe galactose also. All these are examples of monosaccharide molecules. Okay, monosaccharide units of carbohydrate, single unit of carbohydrate. The disaccharides which we are going to see in our video today. So in our experiment, what we are going to see is sucrose, which is a disaccharide. Then we have lactose, and further we have maltose. Okay, so sucrose, lactose, maltose. There is one more difference between sucrose, lactose and maltose, that sucrose is said to be a non-reducing sugar, whereas lactose and maltose are considered to be reducing sugars. We don't have an oligosaccharide in today's today's experiment, but there are many oligosaccharides which you can see and learn about in the theory, like raffinose, then styrose. So different type of oligosaccharides are there. The polysaccharide which we have provided you as a sample today is starch. Okay, but as I mentioned earlier, the other polysaccharides are, let's say, cellulose. Okay, cellulose is a very common polysaccharide which is found in every tree and plant and is responsible for the erectness of trees and plants. There is another polysaccharide, glycogen, something similar to starch, found in animals, okay, in the animal body. So these are example of the polysaccharides, and as I mentioned, starch is the one which we are going to use as a sample today. So that was about the classification of carbohydrates into sugars and complex sugars, why they are sweet in taste and why they are tasteless. I hope we have understood the fact. Let us see the next basis of classification of sugars. So one was based, you can say, based on the number of saccharide units. So based on that, we called them mono, di, oligo and polysaccharide. The next one is based on the nature of sugar, whether they are reducing sugars or non-reducing sugars. When we say reducing or non-reducing sugar, means sugar which is acting as a reducing agent and sugars which act as a non-reducing agent. I'm sure everybody remembers what is a reducing agent. So the one which undergoes oxidation itself and simultaneously helps in reduction of the other component will be called a reducing agent. And in this case, it will be called a reducing sugar, and the one which does not participate or support in the reduction, that will be considered as a non-reducing sugar. Okay, so let us see, and in fact this is one of the basis of the tests that we are going to do today. There are certain tests which give a positive result with reducing sugars, and there are certain tests which do not give a positive test. Okay, so let us we will see about the test after a while, but first let us see the difference between reducing and non-reducing sugars. Okay, so what are reducing sugars? Basically, the reducing sugars are, you can say, in the structure they will have a free aldehyde or a free ketone group, or they will be able to you can say they will be able to modify their structure or change their structure where they get a free aldehyde or a free ketone group. However, in non-reducing sugar, there is absence of a free aldehyde or a free ketone group. When we say free aldehyde or free ketone group leads to reducing sugar, so just now you can recall the structure of glucose and fructose. They have a free aldehyde and a free ketone group. So definitely they will act as a reducing sugar. All these reducing sugars are good reducing agent because they are able to reduce the other component and simultaneously get oxidized. Okay, like for example, I will just write down the reaction here quickly. About how they undergo oxidation and how there is simultaneous reduction. Okay, so again I am drawing the structure of a glucose molecule. As I mentioned before, these are the most this is the most popular type of carbohydrate when it comes to study of the carbohydrate. So this is the structure of a glucose molecule, the aldehyde group. It is this aldehyde group which gets oxidized to the acid. Okay, so this CHO group gets converted into COOH. Everything else remains same. Okay, everything else remains same. Only the aldehyde group gets converted into acid, which is of course oxidation. Okay, so this is oxidation which is happening. I'm sure everybody remembers organic chemistry. So aldehyde to acid, that is oxidation which is taking place, and simultaneous reduction takes place. Okay, reduction of Cu2 plus to Cu plus, cupric ions to cuprous. Okay, so this is reduction. Reduction is taking place. You can see plus 2 becoming plus 1, so reduction, because the the carbohydrate is undergoing oxidation leading to reduction of this species. Therefore we are calling it reducing sugar. This sugar is able to reduce Cu2 plus to Cu plus, and therefore it is reducing sugar. When we say non-reducing sugar, that means the sugar is not able to undergo this type of reaction. In fact, the reaction which I showed you just now uh, takes place in two of the tests. One is Benedict's test and other one is Fehling's test. Okay. So in the video you will also see these tests, and we have mentioned it otherwise also. So we will talk about these tests just after a while. But as of now, for your understanding, the sugars which are able to cause reduction, they are good reducing agents, and the other ones are non-reducing agents, and therefore the term reducing sugar and non-reducing sugar. Okay, we have already discussed they have free aldehyde ketone group. These do not have free aldehyde or ketone group. They give positive test with the Fehling, and non-reducing sugar gives negative test with the Fehling. I told you that this Cu2 plus in the form of copper sulfate is present in the Fehling's reagent, and when it is able to reduce this Cu2 plus ions to Cu plus, then we will say actually there is formation of a red color. Okay, there is formation of a red color, and this red color indicates red precipitate brick red color, and this red color indicates a positive test or a positive result towards Fehling's test. Whereas for non-reducing sugar, it gives a negative test. That means there is the red color does not appear. The brick red color does not appear. So from cupric ion to cuprous ion, the reduction is taking place to give the positive test, and when the reduction doesn't take place, we will call it a negative test. Let us now see which is included in reducing and which is not included in or which is part of non-reducing. So as you can see, it is mentioned here, all the monosaccharides and some disaccharides are reducing sugars. Okay, which either contain a free aldehyde or a ketone group, or they can get converted into a structure containing free aldehyde or a free ketone group. Whereas there are some disaccharides and polysaccharides which do not have a free aldehyde or a ketone group. The examples from the samples that we have today, I'll just mention. So all the monosaccharides, glucose, fructose, okay, these are monosaccharides and are reducing sugars. Amongst the disaccharides, since it's written some disaccharides, so it is very important. Amongst the disaccharide, lactose, which is made up of glucose and galactose, and maltose, which is made up of 2 units of glucose. These are reducing disaccharides. Okay, reducing disaccharides. The other one, the non-reducing disaccharide is sucrose, which we'll be using in the lab today. Sucrose and all polysaccharides, for example, starch. Okay, so starch and other polysaccharides, they behave as non-reducing sugars, and they do not give positive test with the Fehling and Benedict's reagent. So I hope that is clear to everyone. Let us now see what all tests are we going to perform for the carbohydrate. Okay, so actually there is a flow chart based on which we can we can proceed via that flow chart to identify an unknown carbohydrate. So suppose an unknown carbohydrate or an unknown sample is given to you, you will use that flow chart to find out what steps do you need in order to identify the carbohydrate. Okay, so different type of tests are there. I think I will first take you to that flow chart, and then we can come back and see the tests. Yeah. So here is the flow chart. You can see if an unknown solution or a test solution is given to you, how are you going to proceed? The first thing that you will do is the Molish test. Okay, so you will do Molish test, and Molish test is given by all the carbohydrates. In fact, it's the test to determine the presence of carbohydrate. So whether it is a polysaccharide, whether it's a disaccharide, whether it's a monosaccharide, all of these will give a positive test for Molish. If any substance gives a positive test for Molish, you are expected to proceed further to test carbohydrates, otherwise not. Maybe it is some other type of component. Okay, so this Molish test will always be positive. Once you have the positive Molish test, the next thing you need to do is perform Benedict's and Fehling's test. Okay, Benedict's and Fehling's test, if you get a positive result, as we saw just now a while ago in the reaction, that cupric ions are reduced to cuprous ions. So, and it leads to appearance of a brick red color or a reddish color as in Benedict's test. I will just let you know. You can see in the video also. So that indicates a positive test. Appearance of a brick red precipitate or a reddish orangish color in Benedict's indicates a positive test for reducing carbohydrates. Okay. So if the test comes out to be positive, it means the sample is a reducing carbohydrate. If it comes out to be negative, that means it's a non-reducing carbohydrate. We have understood already what is a reducing carbohydrate, and all the monosaccharides and some disaccharides are reducing carbohydrate. Okay, so you take the test solution, you perform the Molish test, you get a positive result. You perform Benedict's and Fehling's, you get a positive result again. The next test that you need to do is the Barfoot's test. Okay. Now this Barfoot's test is to identify amongst the reducing sugar whether it is a monosaccharide or whether it is a reducing disaccharide. Okay. So this Barfoot's test comes out to be positive for a monosaccharide, any monosaccharide, but we have to follow the steps very carefully, especially the heating part, whereas it comes out negative for the reducing disaccharide. Okay, so you will perform Barfoot's test. If you get positive, it means your sample is a monosaccharide. If you get a negative test, that means it is a reducing disaccharide. In case you get this reducing negative test, the last test that you need to perform is Ferron's test. Okay, so this will test the presence of reducing disaccharides. However, if you get a negative test for Benedict's and Fehling, so what will you do over there? That means there is presence of a non-reducing carbohydrate. Non-reducing carbohydrate, if you can recall, it is some disaccharides and all polysaccharides. So next what we are going to do is the iodine test. Iodine test, what is observed? If you get a positive iodine test, that means it is polysaccharide. A positive iodine test, that means appearance of blue color. Okay, with starch for that matter, that means it is a positive test for the polysaccharide. However, if iodine test comes out to be negative, then definitely it is a non-reducing disaccharide, the example of which is sucrose. Okay, so this is the flow chart that you use to proceed if an unknown, or if you have to make a test and you have to identify which type or which class of carbohydrate it is. I hope this was clear to everyone. Let us now quickly see the tests, the various type of tests. Okay. So Molish test, as I was saying, Molish test is positive. It should come out to be positive for all carbohydrates. Okay. Positive for all carbs, all type of carbohydrate. Okay, so you can see over here, this is the Molish reagent, 5% alpha naphthol solution in ethyl alcohol, and basically you take the test solution and you add Molish reagent to it, and from the side of the test tube you put concentrate sulfuric acid. Appearance of a purple colored ring at the junction of the two liquids indicate a positive Molish test. Okay, it is expected that all carbohydrates, whether mono, di, oligo or poly, is going to give a positive test for Molish. What you can see in the picture over here is the test for Benedict's reagent, or you can say test with Benedict's reagents, Benedict's test as we call it. This Benedict's test is also semi-quantitative in nature. Okay, though the identification tests are mainly qualitative, but this Benedict test you can say is semi-quantitative in nature. Why? Because the color of the solution, or the you can say the precipitate, indicates the amount of reducing sugar which is present in the sample. Okay, so as you can see, if a brick red color appears, large amount of reducing sugar in the sample. If you get an orange red precipitate, little less than the maximum, that means moderate amount of reducing sugar. And then if you get a greenish or yellowish PPT, then that means there are traces of reducing sugar. If the color doesn't change at all, that means it does not contain any reducing sugar. Okay, so that is why we say Benedict's test is a test to identify the reducing sugar. You can see the components of the reagent which are involved, which is involved in the Benedict's test. Okay, so made up of copper sulfate, that's why blue in color, and sodium citrate and sodium carbonate to make more alkaline, sodium citrate to provide citrate ions and make the solution little more stable. The next test is Fehling's test, and Fehling's test is another important test for testing the presence of reducing sugar. Okay, so in this case, again what happens, the same reduction takes place from Cu2 plus to Cu plus, cupric to cuprous. Okay, the difference between Fehling and Benedict's is that Fehling has two solutions, Fehling A and Fehling B. Okay, this Fehling A and B needs to be mixed just before you start the test. Okay, Fehling A and B. So Fehling A is a copper sulfate solution and Fehling B is a solution of sodium potassium tartrate, which is also called Rochelle's salt. So when you mix them together, a dark blue ink blue color will appear, and to that we mix it with the test solution, ultimately giving us a brick red color. So if you see the appearance of a brick red color, that means it is a positive Fehling's test, and if you do not see the appearance of this color, that means it's a negative Fehling's test. If you test negative for Fehling, what do? You do the iodine test. I'm sure everybody must be remembering the iodometric titrations which you might have done, and many places we use starch as the indicator. So basically that is the same test, we are kind of same reaction we are doing. Starch and iodine react together to give a blue-black color complex. Okay, so this test is specific for the polysaccharides, and in fact this test is used to differentiate between polysaccharides and the remaining carbohydrates. Okay, so starch in this case gives a blue color, a deep blue or blue-black color with the iodine. So if your Benedict's and Fehling's go negative, you do the iodine test. If you get a positive result, it means it's a polysaccharide. If you get a negative result, then that means it's a non-reducing sugar. The next test is Barfoot's test, and if you can recall, if Feheling and Benedict come out to be positive, the next test you must perform is the Barfoot's test. What is there in the Barfoot's test? What do? You actually identify or distinguish between the monosaccharides and the disaccharides. So this test basically helps in identifying whether the sample is a reducing monosaccharide or it is a reducing disaccharide. Okay, so again in this color, the method you have to be very careful. The heating needs to be done 2 to 3 minutes only, and the appearance of light red precipitate, which is kind of brick red only but very small in quantity, when it appears, that means it is a positive Barfoot's test. Any other color, it is not a positive test at all. The Ferron's test, as we mentioned, if we get a negative Barfoot's test, ideally we should do a Ferron's test. But since it was not available in the video, we have not put it in the, not available in the lab, so we have not put it in the video. However, you may find several videos of Ferron's test also in the YouTube. Now what I'm going to do is the link, you can see, for the method, for the purpose of method, we have made a video of how we are going to perform these tests, whether it's Smallish, Benedict's, Feheling's, Barfoot's and iodine test, and this link I will put separately in the chat box or in our as a post, and you can click on this video and you can watch this video. Once you have seen the video, and if you have any questions, you can make a list of your questions, and then we can have a question answer session based on this video. Okay, so the next what we are going to do is we will watch this video and you can come up with any questions or queries that you have about the method that we are going to use in the going to show in the video. Finally, you may have all watched the video, and finally we were able to identify the reducing and non-reducing sugars from our sample. So different samples were there: glucose, fructose, lactose, maltose, galactose. All these are considered to be reducing sugars. Monosaccharides, all of them are reducing. Disaccharides, some of galactose is also a monosaccharide. So lactose, maltose, it's a reducing disaccharide. Sucrose is a non-reducing disaccharide, and starch, of course, is a polysaccharide. Okay, so I guess that is all for today's class, and you can see some additional reading material over here which can explain the health benefits and risk associated with using or intaking carbohydrates in our diet. Okay, so that is all for today's class. Once we have done this, we are going to have a small quiz after this. Thank you everyone. Thank you one and all.